Hi everyone! In this video, we are going to talk about Cell Cycle Part 2, a continuous topic presented by previous group. This video is brought to you by Group 14 with dedication of Siti Aisyah binti Sohimi, Muhammad Faik Saifullah bin Kamaruddin, Aina Aisyah binti Muhammad Aminun, and also Ng Yong Hee. So please enjoy this video. This video has two learning outcomes, and at the end of this video we hope that you are able to describe the intrinsic and extrinsic pathway in details, and secondly able to describe the regulation of intrinsic and extrinsic pathway using an examples. There is two major types of apoptosis pathway, which are intrinsic pathway, also known as mitochondria pathway, and extrinsic pathway. So intrinsic pathway is stimulated from inside the cell in response to stress development signal. This pathway also involved in autocrine signaling, where the cell will release signal to themselves. So for extrinsic pathway is triggered by extracellular signal protein binding to cell surface their receptor. So extrinsic pathway involved in parakine signaling, where the cell will release signal to neighboring cells. Next, I will explain intrinsic pathway in details. So intrinsic pathway is regulated by members of BCL2 family protein. This is because some BCL2 family protein are anti-apoptotic while some are pro-apoptotic. So anti-apoptotic protein will inhibit apoptosis by blocking the release of cytochrome C, while pro-apoptotic will promote apoptosis by enchanting the release of cytochrome C into the cytosol. So this pro-apoptotic has two subfamilies, which are the effectors BCL2 family protein and BH3 only proteins. So the figure below show the examples of anti-apoptotic and pro-apoptotic BCL2 family protein. In inactive intrinsic pathway, which means the absence of apoptotic stimulus such as stress and DNA damage, the anti-apoptotic BCL2 family protein will bind to and inhibits the effector BCL2 family protein. So in these situations, no cytochrome C will be released, thus no apoptosis occur. So in active intrinsic pathway, which means in the presence of apoptotic stimulus, BH3 only protein are activated and it will bind and inactivate anti-apoptotic BCL2 family protein, which will allow the aggregations of pro-apoptotic effector, for example Bach and Bats, on the surface of the mitochondria. So this situation will cause the release of cytochrome C and subsequent apoptosis inductions. So the mechanism of intrinsic pathway will next be explained in detail by Faik.
Okay, next I will present to you about the regulation of intrinsic pathway. So please watch this video.
The intrinsic pathway is also called as the mitochondrial pathway, owing the essential involvement of mitochondria. Mitochondria consists of two layers, which are outer and inner membrane, and the space between these two membranes is called as intermembrane. Cytochrome C and the other proteins are located inside the intermembrane space. In the apoptosis case, when Bach or Bach's proteins are activated by apoptotic stimulus such as stress, they aggregate in the outer mitochondrial membrane, leading to the release of cytochrome C into the cytosol. Cytochrome C then binds and activates the adaptor protein, which then assemble to form the seven-armed complex called the apoptosome. This complex leads to the recruitment of the initiator procaspase, specifically called procaspase 9. The activation of procaspase 9 within the apoptosome will activate the executioner procaspase. This active executioner caspase leads to a caspases cascade, then apoptosis will occur.
Okay, next I will explain about the DNA damage and apoptosis. So I will explain to you about the DNA damage can temporarily halt progression through G1 checkpoint. The cell cycle control system uses several distinct mechanisms to halt progress through the cell cycle if DNA is damaged. The mechanism that operates at the G1 checkpoint will prevent the cell from replicating the damaged DNA. DNA damage in G1 phase will cause the increasing of concentration of p53 protein. p53 protein then activates the gene encoding a CDK inhibitor protein called p21. p21 protein then binds to SCDK to prevent them from entering the S phase. The arrestment of the cell cycle in G1 phase gives time to the cell to repair the damaged DNA before replicate in the S phase. If the DNA damage is too severe to be repaired, the p53 protein will induce the cell to undergo the apoptosis. After that, I will explain to you about the extracellular survival factor suppresses apoptosis. There are two ways. Based on the diagram A, the apoptosis will be inhibited when the BCL2 family members are being regulated. In this case, the survival factor binds to the receptors that activate the intracellular signaling pathways. Then, it will activate the transcription regulator in the cytosol, thus inhibits the apoptosis when the gene encoding BCL2 is activated. In the meanwhile, based on the diagram B, the survival factor binds to a cell surface receptor protein which activates the intracellular signaling pathway. This pathway leads to the activation of protein kinases B, also known as AKT kinase, that phosphorylate the BET protein. BET promotes apoptosis by binding to and inactivating apoptosis inhibitor proteins such as BCL2. Once phosphorylated, BET releases BCL2 protein which act to inhibit apoptosis. Thus, the survival factor suppresses apoptosis by keeping BET phosphorylated and out of action.
So now I will explain about the extrinsic pathway. This pathway can be induced by extracellular signals, which is the death ligand from neighboring cells bind to the death receptors. Here are some examples for death receptors with its complementary death ligand, which are FAS, TNFR1, APO2 or DR5, APO3 or DR3. So this is the example for the extrinsic pathway. The FAS ligand from neighboring cells bind to the FAS, which is the death receptors.
How FAS death receptor trigger the extrinsic pathway of apoptosis? FAS death receptor is present on the cell surface. It has a trimeric extracellular ligand binding domain and intracellular death domain. The FAS death receptor is activated when the FAS ligand on the surface of a killer lymphocyte bind to the extracellular ligand binding domain. Then, the FADD adapter protein will bind to the intracellular death domain. The death effector domain of initiator caspase-8 will bind to the death effector domain of FADD protein. The recruitment of initiator caspase-8 and FADD protein forms a death-inducing signaling complex, DIS. Within the DIS, two adjacent initiator caspases interact and cleave one another to form an activated caspase-8 dimer. This stabilizes and releases the activated caspase-8 into the cytosol. The activated caspase-8 will then activate downstream executional procaspases by cleaving them. Eventually, apoptosis is induced.
We also can link the apoptosis to UPR, unfolded protein response, which is in the lecture 11 and 12, protein sorting and transport. The UPR can be found in the quality control in the endoplasmic reticulum lumen. The accumulation of misfolded proteins in the ER will trigger an unfolded protein response. The UPR allows a cell to adjust the size of its ER according to the load of proteins entering the secretory pathway. However, if an expanded ER cannot cope the number of misfolded proteins, the UPR will direct the cell to self-destruct by undergoing the apoptosis. The UPR will trigger intracellular signals as an apoptotic signals to activate the intrinsic pathway. So what is the difference between the cell division and apoptosis? The cell division will increase the number of cells, while the apoptosis will decrease the number of cells.
So this is a diagram that summarizes and compares the extrinsic and intrinsic apoptotic pathway. The two pathways is different in the initiation and caspase activation phase, but both pathways trigger the downstream executional caspases by the initiator caspases followed by apoptosis. What I would like to highlight here is, in some cells, the extrinsic pathway is linked to the intrinsic pathway. Why? It proposes to amplify the caspase cascade to kill the cell. And in this case, the BID protein or BID protein acts as a bridge that connects the two pathways. Normally, the BID protein is inactive and it will activate by caspase-8 in certain cells. The activated BID protein will translocate to mitochondrial membrane. Then it will inhibit anti-apoptotic BCL2 family proteins, therefore amplifying the dead signal to trigger intrinsic pathway. That's all for our presentation. Thank you.